We're now ready for video number 4 in our video series covering energy for life. In video 3, we talked primarily about stages 3 and 4 in the complete breakdown of a molecule of glucose or carbohydrate. Stage 3, you remember, is the Krebs cycle. And again, it's referred to as a cycle because OAA is both one of the beginning substrates, but it's also the last end product in the cycle of the Krebs cycle. We go through several important redox reactions during the Krebs cycle, and in 3 of them, we reduce the coenzyme NAD to NADH. In the 4th, we reduce the coenzyme FAD to FADH2. Those can then transport those electrons to electron transport, where they'll be used to build a proton gradient. We also directly phosphorylate ADP to ATP. And then finally, we continue with the breakdown of the backbone of the pyruvate molecule in the form of carbon dioxide. And that carbon dioxide, again, is transported to your lungs where you breathe it out. And so in summary, one turn of the Krebs cycle, sending one acetyl-CoA through the Krebs cycle, results in the formation of 3 NADH, 2 CO2, 1 FADH2, and 1 ATP. Stage 4 is electron transport and oxidative phosphorylation. In this particular diagram, the flow of electrons is now represented by the blue arrow, and you can see here NADH again is donating electrons to complex 1, whereas FADH2 comes in a little bit later and donates electrons to complex 2. But from there, the electrons get passed on to coenzyme Q10, to complex 3, to cytochrome C, and finally to complex 4, before ultimately they're picked up by oxygen or the final electron acceptor. And again, you know that the purpose of electron transport is to generate a proton gradient. And so we use the energy from these redox reactions to power proton pumps. And these proton pumps are going to pump protons from the mitochondrial matrix into the intermembrane space. And so you can see that that will happen several times where we pump these protons up. And so what we do is we create this very strong proton gradient. Now, that proton gradient is used. It's very useful because as the protons fall down the proton gradient, we direct them through the respiratory assembly, and we use that to do chemical work. We do the chemical work of phosphorylating ADP to ATP. And again, don't forget that the enzyme that allows this to happen is ATP synthase. And so now we've gone through the complete breakdown of a molecule of glucose, including stages 1, 2, 3, and 4. And here's a slightly different overview of that whole process. Here we see stage 1, or glycolysis. More specifically, it's a slow glycolysis, and we know that because we end up with pyruvate. We don't really see stage 2 here, but as the pyruvate is moved into the matrix of the mitochondria, it does get converted into acetyl-CoA. And then we go into stage 3, and then finally stage 4. And again, you understand that stage 4 is where the majority of the ATP is produced. This is a slightly different overview of the 4 stages. And here again is slow glycolysis, where we get our 2-pyruvate. Now, pyruvate can be converted into a number of things, and as we've already seen, 2 of those things include lactate and acetyl-CoA. If we're going to go through fast glycolysis, the NADH are going to give their electrons back to the pyruvate, converting it into lactate. On the other hand, if we're going to continue with the aerobic breakdown of glucose, the pyruvate have moved into the mitochondria, where they then get converted into acetyl-CoA. And then we go through stage 3, and then finally stage 4. Now, I've told you several times that I want you to memorize the overall reaction for cellular respiration. And let's check and see how you're doing here. So pause the video and write that down. Now, if you're still struggling with it, this diagram should help you to be able to do that. And you know that cellular respiration, we begin with glucose. Well, there it is, C6H12O6. And we require oxygen in order to be able to break it down. and it requires 6 O2s. So there are our beginning substrates, glucose and O2. We break down the backbone of the glucose and we get CO2, 2 from stage 2 and 4 from the Krebs cycle. And then finally we produce 6 waters and we get our ATP. And so there is the overall equation for cellular respiration. Here's another overview of cellular respiration that you should be able to kind of annotate or walk your way through, beginning with the glucose being up here in the capillary. It has to pass through the interstitial fluid, and then it gets into the cell via facilitated diffusion using the GLUT molecule. And so then from here on, you should be able to kind of talk your way through the rest of this. You do need to give more detail here for glycolysis. Remember, glycolysis involves activation, cleavage, and the final pathway. Here's stage 2, talk your way through that, and then stage 3 and stage 4. And if you're still struggling with this a little bit, then it's important that you get in to see me so that we can talk about it. Or if you're taking this on a different campus, get in to see your professor and get the help that you need so that you understand what's going on. Now, it's true that we can use fuels other than carbohydrate to regenerate ATP. And in fact, we can use fats as well. Most of our energy to replace ATP comes from the breakdown of carbohydrate and from fats. And we have fatty acids, but fatty acids do not go through glycolysis. Only glucose goes through glycolysis. So the first stage in the complete breakdown of a fatty acid is referred to as beta oxidation. And we'll look at beta oxidation in just a minute. But from there, we then go through the Krebs cycle just as we did with carbohydrate. And finally, we go through electron transport and oxidative phosphorylation. Again, just as we did with our carbohydrate. Now, I don't want you to get too worried by what you see here. I just want to point something out real quickly. And so, as you look at this, you should notice that we have this molecule that's being broken down in the presence of oxygen, and it results in the production of CO2, water, and ATP. And that's true for all three of these things. And so what you should say is, oh, this is cellular respiration, and it happens to be cellular respiration of fatty acids. So again, you don't need to memorize this, but when you see that something is being broken down in the presence of O2 and we're producing CO2, water, and ATP, you should automatically think, oh, this must be cellular respiration. The other thing I'd like you to notice is that this first fatty acid has 16 carbons in it, and we produce 16 CO2s. This fatty acid is longer. It has 18 carbons and we produce 18 CO2s. And then finally we have an even longer fatty acid with 20 carbons and we produce 20 CO2s. And so when we completely break down a fatty acid, all of the carbons in the backbone of that fatty acid are going to be converted to CO2. That CO2 is going to be transported to your lungs and you're going to breathe it out. Okay, so as we've already said, most of our energy for replacing ATP comes from oxidation of carbohydrate and lipids. And the majority of the lipid that you have in your body is in the form of triglycerides. And here is a triglyceride, which you should recognize from our discussion of chemistry. Here's this 3-carbon backbone, which is a glycerol molecule. And then we've attached to that 1, 2, 3 fatty acids, hence triglyceride. Now, before we can go through a beta oxidation with our fatty acids, we have to break the fatty acids off of the glycerol molecule. And this process is referred to as lipolysis. And again, you know lysis means breakdown, and lipo refers to lipids. So lipolysis is the breakdown of these fatty acids. Now the fatty acid can move into the mitochondrial matrix where it will go through beta oxidation. Here's an overview of beta oxidation. And again, up at the top, we have our triglyceride. And so the first thing that we have to do is we have to go through lipolysis to break the fatty acids off of the triglyceride. Now the fatty acids can go ahead and go through beta oxidation. And here's a little bit of a schematic representation of that. And you look at what's happening right here. You notice that the very first thing that happens in beta oxidation is that we use energy from ATP to phosphorylate the fatty acid. Does that sound familiar to you? I hope that it does, because that's exactly what we did to glucose. Remember, the first part of glycolysis is activation. Well, now we take that activated fatty acid and we go through some redox reactions. We get NADH, we reduce FAD to FADH2, and then we chop off a 2-carbon fragment, which is in the form of acetyl coenzyme A. And so there we go. We break off 2 carbons at a time, forming acetyl coenzyme A, and we also go through redox reactions in which we produce both NADH and FADH2. Well, what's going to happen to the acetyl coenzyme A? As you can see here in the diagram, the acetyl coenzyme A goes into the Krebs cycle. What is it that's going to happen to the NADH and the FADH2? Well, the FADH2 and the NADH are going to go to electron transport. Now, what this diagram doesn't show you is that after we've clipped off the 2-carbon fragment, we still have a fatty acid that can be broken down even more. And so that piece comes up and goes through again. It doesn't need to be activated again. We've already activated it. Only happens once. It doesn't happen more than that. But we do go through redox reactions again, and we get more NADH, more FADH2, and more acetyl-CoA. And so this diagram shows you a little bit more detail in terms of what's going to be happening here. If you count these, you'll see 2, 4, 6, 8, 10, 12, 14, 16 carbons. And so we're actually going to go through the steps of beta oxidation 7 times. And as a result of that, you can see, look, we're going to get 8 acetyl-CoA. We don't just get 1 or 2, we actually get 8. And the number of acetyl-CoA that are formed is easy to determine. Count the number of carbons, divide by 2, and that will tell you how many acetyl-CoA that you get. If the fatty acid has 22 carbons, you get 11 acetyl-CoA. The number of NADH and FADH is actually 1 less than that. And so to determine the number of NADHs and FADHs, count the carbons, divide by 2, but then subtract 1. And so now here you can see that what we're going to do is we're only going to get 7 NADHs and FADHs from this 16-carbon fatty acid. And the reason for that is each time we go through and snip off 2 for acetyl-CoA, the fatty acid gets a little bit shorter. So we do 1 cut, then we're down to 14, now we're down to 12, to 10, to 8, to 6, and finally we're down to 4 carbons that we have here. I made the cut a little bit too deep. And so here we've got 4 carbons left. 1, 2, 3, 4. And we're on our 7th time through. We're going to cut this in half, and each of them will become an acetyl-CoA. And so that's why we get 8 acetyl-CoA, but we only get 7 NADHs. This is a slightly different depiction of beta oxidation. And again, here you can see now we have an 18-carbon fatty acid, and they're highlighting these 2 on the end, which are going to be clipped off and become acetyl-CoA. And so again, the first thing that we have to do is activate it. And then we're going to go ahead and we're going to cleave it. We go through our redox reactions. And when we do that, we get NADH that goes to electron transport and FADH that goes to electron transport. That NADH will move enough protons into the intramembrane space to power phosphorylation of 2.5 ATP. The FADH will move fewer protons, and so we can only phosphorylate 1.5 ATPs. And then finally, our acetyl-CoA is formed and it goes into the Krebs cycle. And so again, remember that now this 16-carbon fatty acid is going to come up and go through this again. And then we'll be down to 14. And then it will come up and go through again. We'll be down to 12, and we'll continue to go through that until we've gotten 9 acetyl-CoA, and we've gotten 8 NADH and 8 FADHs. And so there, we've gone through cellular respiration for both carbohydrate and lipid.